Hello, my name is Oliver and I work at the Ontario Institute for Cancer Research. And today I'm going to be discussing my master's thesis project, which is titled Characterizing the Genome-wide Associations of Somatic Mutations and Chromatin Accessibility in Cancer. So we know that cancer is a disease caused by somatic mutations, and mutations which are positively selected for and confer oncogenic fitness to cells are considered cancer drivers. So if you look at the figure on the right, we see the timeline of tumor evolution started by a normal cell, which acquires a driver mutation, leading to it becoming the founder clone. And as the tumor acquires more driver mutations, more subclones develop with different oncogenic properties, such as the ability to metastasize, as we can see here. However, most mutations in cancer cells are actually neutral passengers, and these can occur due to a variety of mutational processes. Interestingly, mutations are not distributed equally throughout the genome. We know that mutations occur due to a complex interplay between DNA damage and repair processes, but different processes occur at different scales. So if we look at the smallest scale, we see at the scale of transcription factor binding sites, localized increases or spikes in mutations at these transcription factor binding sites. And if we look at the largest megabase or million base scale, we see regional increases in mutations at later replicating closed chromatin domains. So mutational processes actually generate characteristic mutational signatures, which can be mathematically deciphered. And a minority of these signatures are actually attributed to known mutational processes. So this is what a signature looks like here on the right, which is just a linear combination of different mutation types based on the actual mutation and its trinucleotide context. And if you look at single base signature 4, which is attributed to smoking, we see the enrichment of C to A mutations. While if you look at for signature 7A, which is attributed to UV light damage in skin, it's related to C to T mutations. You can actually go into a catalog of mutations within a tumor sample and attribute specific signatures to each mutation to understand where those mutations are coming from within the tumor. So just as an overview of my project, for our data, we have primary tumor, normal cancer, normal cell line, whole genome chromatin accessibility, and replication timing data available from the TCGA roadmap and ENCODE project, over 600 total tracks or samples. And we also have whole genome sequencing mutation data for over 2,500 primary tumors from the PEACOCK project. And the aims of my project are to first evaluate whether cancer type specific regional mutation frequencies are better represented by the chromatin landscapes of tumor cells or normal cells. And the second aim is to uncover the association between specific mutational processes occurring in cancer cells and the chromatin or replication timing landscape. And this is the overview of the methodology of my project. We start with whole genome somatic mutation tracks, as well as whole genome chromatin accessibility and replication timing tracks. And what we do here is we split the genome into mega base size or million base size windows. And for our mutations, we take the total sum of mutations overlapping that window within a specific cancer type. And in this way, we create a regional mutation variation track for a specific cancer type. And we do something similar for our chromatin accessibility and replication timing profiles, where for each window, we take the average chromatin accessibility or replication timing for that tissue sample or tumor sample to build a regional variation of chromatin accessibility genome-wide for the sample. And then what we do is we train a random forest machine learning model on our chromatin accessibility and replication timing tracks at the megabase scale to predict regional mutation frequency at the megabase scale of a specific cancer type. And then we can look at the accuracy of this model to see how associated chromatin accessibility from these tissues are with regional mutation frequency of this cancer type. We can also look at predictor importance to see which of these tissues or tumors were most predictive of regional mutation frequencies in this cancer type. And as our first analysis, we want to understand whether tumor chromatin accessibility or normal cell chromatin accessibility is more predictive of regional mutation frequencies in different cancer types. So these bar plots represent this result. And so on the x-axis, you see the different cancer types that we did this analysis in, which is 25 different ones in pan cancer. And on the y-axis, what you see is the increase in model accuracy of the model trained on tumor chromatin accessibility in relation to the model trained on normal chromatin accessibility. And what we see here is that in most cancer types, the tumor chromatin accessibility model is actually most predictive of regional mutation frequencies. However, in a few cancer types, it's actually the normal cell chromatin accessibility model that's more predictive. And these include medulloblastoma, melanoma, leukemia, and lymphoma. And what this implies that in these cancer types, we're we might be looking at earlier mutational timing within tumor evolution because it's a normal cell chromatin accessibility that's most associated with their mutational landscape. What we can do also is to analyze which tissues or tumors were contributing the most predictive chromatin accessibility or replication timing tracks to predicting regional mutation frequencies in these cancer types. So we take melanoma as an example. By far and away, the most predictive chromatin accessibility track comes from a normal skin melanocyte, which is known to be the cell of origin for melanoma. So we see these cell of origin or tissue of origin relationships. What we also see is that the most predictive chromatin track is actually coming from the normal skin melanocyte as opposed to a melanoma tumor chromatin accessibility. And what this implies is that the mutations in melanoma tend to occur earlier within tumor evolution because they're most associated with a normal cell's epigenome. And then we can extend this analysis to 14 cancer types within our study that actually had chromatin accessibility or replication timing tracks from similar tissues. And what we find is that in most cancer types, the most predictive tracks are coming from matching tissues or potential tissues of origin. So this is happening in 12 out of 14 cancer types. And then even in the ones where we see coming from a different tissue, such as an esophageal adenocarcinoma, where the most predictive chromatin accessibility tracks are derived from stomach adenocarcinoma, there's evidence in the literature that the cell of origin of these two cancer types are very related. What we also see is that in many cancer types, the most predictive chromatin track is derived from a primary tumor, indicating later mutational timing. And we see this in breast adenocarcinoma, kidney cancer, liver, colorectal, stomach, and lung adenocarcinoma. However, in some cancer types, it's actually derived from normal cells, indicating earlier mutational timing. And we see this in lung and head squamous cell carcinoma, with normal squamous cell replication timing being the most predictive. And this implies possible early mutational timing because of exposure to tobacco smoke prior to oncogenesis. And then we can we see a similar story here with lymphoma and leukemia, where it's the normal B cells are the most predictive, as well as in melanoma, where the normal skin cells, as I previously mentioned. Interestingly, in glioblastoma, the most predictive chromatin tracks either come from normal brain cells, such as those of the hippocampus, astrocytes, and spinal cord, as well as from lower grade glioma cells, which are brain tumors that are an earlier step in tumor evolution in some glioblastoma tumors. Again, implying possible earlier mutational timing in this cancer type. What we can also look at is how these chromatin accessibility tracks are contributing to the model output, which is predicting the number of mutations within a window. And what we see here are violin plots or density plots of how these chromatin tracks are contributing to the SHAP score, which is the model output. And what we see here is that when melanocytes are in open chromatin, which is red, we see them contributing decreased mutations to the model. When they're in closed chromatin, which is blue, they're contributing to increased mutations to the model. And this is consistent with our understanding that open chromatin regions tend to accumulate less mutations and closed chromatin regions tend to accumulate more mutations. And the next step that we wanted to look at is what the relationship between specific mutational processes and the chromatin landscape is. So what you see here are different signatures or processes split by their group of mutational process, such as carcinogenic signatures, defective DNA repair, and so on. And on the y-axis, what you see here is the model accuracy of predicting regional mutation frequencies by chromatin accessibility tracks of these specific signatures. So essentially their signatures association with the chromatin or replication timing landscape. And the first thing that we found is that carcinogenic mutational signatures are significantly more associated with the chromatin landscape than those derived from endogenous mutational processes, such as defective DNA repair, APOBEC enzyme activity, and cytosine deamination in signature 1. We also found that in several unknown signatures of unknown etiology are also significantly more associated with the chromatin and replication timing landscape than endogenous mutational processes. And I wanted to highlight signature 17 in esophageal adenocarcinoma. There's been previous evidence in the literature that signature 17 actually occurs due to reactive oxygen species from gastric acid reflux in the esophagus. And because we see its high association with the chromatin and replication timing landscape, we have further evidence that this could be also a carcinogenic signature that's coming from a carcinogen that's coming from outside the cell. And then if we examine SHAP score for specific signatures in specific cancer types, we can look at signature 5 and signature 13 in breast adenocarcinoma. Signature 5 is of unknown etiology, but it is clock-like, meaning that it increases with age. And we see that the most predictive chromatin track of this signature in this cancer type is derived also from breast adenocarcinoma. But importantly, open chromatin regions we see are depleted in mutations, while closed chromatin regions are leading to an increased mutation rates within our model, which is expected. But in signature 13, which is related to APOBEC enzyme activity, we see that actually open chromatin regions are contributing to increased mutation rates within our model. And there has been previous evidence in the literature that APOBEC enzyme activity is increased in open chromatin regions, and therefore this leads to increased mutation rates from this specific mutational process in open chromatin regions. And it's really interesting that we see this come up also within our analysis. So just to conclude, we found that chromatin accessibility profiles derived from primary tumors are better predictors of regional mutation frequency than those derived from normal cells in most cancer types. The exceptions include melanoma, leukemia, lymphoma, and glioblastoma. We found that cancer mutation rates tend to be predicted by chromatin accessibility or replication timing derived from the same tissue, whether it's normal or tumor. We also found that carcinogenic mutational processes are highly associated with chromatin accessibility and replication timing, while endogenous signatures are less associated. And as I highlighted in the previous two slides, first, signature 17 of unknown etiology in cancer is inferred to be of exogenous origin to the cell, namely gastric acid reflux. And finally, APOBEC enzyme activity mutations are increased in open chromatin. So I just wanted to acknowledge everyone from my lab and my supervisory committee, and thank you so much to the audience for listening.